What is genetic linkage? Genetic linkage is the tendency of DNA or genes sequences that are close together on a chromosome to be inherited together during meiosis phases of sexual reproduction. Some genes are always inherited together with other genes because they are on the same chromosome or they are linked. So, when this particular chromosome is passed into a gamete, all the genes on that chromosome can also pass into that gamete. Peterson and Punnett were the great scientists or eminent scientists who investigated the inheritance of linked genes. These two scientists conducted their research on flower color and pollen shape on sweet peas. In their experiment on sweet peas, in case of flower color, there can be two contrasting features, purple flower color or red flower color. In case of pollen shape, there can be two contrasting features like round pollen shape or long pollen shape. The alleles are capital P that determine purple flower color, which is dominant. Small p determines red flower color, which is recessive. Capital L determine long pollen shape, which is dominant. And small l that determines round pollen shape, which is recessive. Here, capital P and capital L are on the same chromosome, and small p and small l are on the same chromosome, which means capital P and capital L are linked, and small p and small l are linked. We can show the genotype as two sets of this unit as capital P capital L slash capital P capital L. Capital P capital L before slash indicated that these two alleles are found on the same chromosome, whereas capital P capital L after slash indicates that these two alleles are found on the same chromosome or they are linked. Because they are linked, the two genes are inherited as a single unit. The phenotypes that can be determined by capital P capital L slash capital P capital L is purple flowered with long pollen, and the phenotypes that can be determined by small p small l slash small p small l is red flowered with round pollen. When we cross two parents whose genotype is capital P capital L slash capital P capital L and small p small l slash small p small l, their probable offsprings could be capital P capital L slash small p small l, capital P capital L slash small p small l, capital P capital L slash small p small l, capital P capital L slash small p small l. When we see phenotypic and genotypic ratio, phenotypic ratio all are purple flowered and long pollen shaped. Genotypically, all are capital P capital L small p small l. When Bettison and Punnett led heterozygous strains of F1 generation to self-fertilize or self-pollinate, their probable offspring could be capital P capital L slash capital P capital L, capital P capital L slash small p small l, capital P capital L slash small p small l, small p small l slash small p small l. When these scientists led heterozygous strains of F1 generation to self-fertilize or self-pollinate, They also found other probable offsprings. These probable offsprings could be capital P capital L slash small p capital L, small p capital L slash small p small l, capital P capital L slash capital P small l, capital P small l slash small p small l. After self-fertilization of F1 generation, Peterson and Punnett phenotypically acquire in F2 generation 284 purple flowered and long pollen, 21 purple flowers, round pollen, 21 red flowers, long pollen, and 55 red flowers, round pollen that couldn't show a 9:3:1 ratio of dihybrid inheritance. In the previous moment, we studied that purple flower color goes with long pollen shape and red flower color goes with round pollen shape because the two genes are behaving as a single unit or they are linked. That means they found on the same chromosome. Peterson and Punnett would not predict any plants that had either purple flowers with round pollen or red flowers with long pollen. You see, now purple flower goes with round pollen shape and red flower goes with long pollen shape. How can this be happen? Here we must not forget that when these gametes are formed, they are formed by meiosis and that this involves crossing over at prophase 1. The consequence of crossing over at prophase 1 is some gametes do contain the combination capital P small l and others contain small p capital L. So we do get the four types of offspring from a cross between two plants, both heterozygous for both features, but not in a 9:3:1 ratio. Crossing over between two homologous pairs or bivalents during prophase 1 led recombinant types to occur that we would not expect from a linked inheritance. Much of the early research on crossing over and recombination was carried out using fruit flies. Why fruit flies were selected as convenient experimental animals? Because fruit flies or Drosophila melanogaster are small with short life cycle. They live just about 2 weeks. Second, fruit flies are cheap and easy to breed and keep in large number. Third, they have only 4 pairs of chromosomes per cell. Fourth, chromosomes of fruit flies are large enough and easily be observed. And fifth, staining of chromosomes reveal dark bands which corresponds to particular genes. In early 90s, Thomas Morgan was able to prove that genes carried on chromosomes are the physical basis of inheritance, and his students Sturtevant experimented on linkage and crossing over in Drosophila melanogaster to make predictions about what offspring to expect if there was no crossing over. Sturtevant expected fruit flies that have gray body, red eyes, and fruit flies which have black body, purple eyes. But due to crossing over or recombinant formation, he obtained or acquired a small number of fruit flies that have black body, red eyes, and small number of fruit flies which have gray body and purple eyes. As I reminded you before, gray body goes with red eyes and black body goes with purple eye color. That means alleles for gray body and alleles for red eyes are linked, and alleles for black body and alleles for purple eyes are linked. But due to crossing over or recombinant formation in meiosis 1 at prophase 1, alleles capital G for gray body, which was linked with allele capital R for eye color, goes with small r that determine purple eye color. If gametes of black body purple eye, that is small g small r and small g small r, randomly fused with gametes of gray body red eyes, capital G capital R and small g small r, half of the offspring could be gray body red eyes and half of the offspring could be black body purple eyes. If gametes of black body purple eyes, that is gamete small g small r and small g small r, randomly fused with the recombinant gametes of gray body red eyes, which is small g capital R and capital G small r, half of the offspring could be black body red eyes and half of the offspring could be gray body purple eyes. Recombinant individuals of fruit flies, which are black body red eyes and gray body purple eyes, occurred in fruit flies population due to crossing over. Then he counted the number of expected and recombinant types to find the percentage of recombinant types or the crossover value. COV or crossover value is equal to number of recombinants over total number of offsprings times 100. This crossover value is a measure of how far apart the genes are on a chromosome and hence from the center of the chromosome. A low crossover value or frequency indicated that the genes are close together on a chromosome and close to the center of the chromosome. A high value indicated that the genes are farther apart with each other and from the center. So, a chance of crossing over occurring between two genes depends on how far apart they are on the chromosome and hence from the center of the chromosome. If genes are very close together, then it is unlikely that crossing over will occur between these genes. If they are farther apart, it is more likely that crossing over will occur between them and that recombinant types will be formed. This has been used in creating gene maps of chromosomes, where the percentage of crossover values are used as a unit of separation of the genes on a chromosome. So gene mapping or linkage mapping is a map that shows the spatial arrangement of genes on a chromosome. Gene mapping has been used to track down genes that can cause genetic disease or genetic disorder so that the DNA of the gene can be cloned for analysis and research.